Hello friends, Ace Botany next episode. In this episode, we'll be discussing Cell, the Unit of Life, 7th part, last part. Now in first year, only remaining chapter is Biological Classification. Second year, only remaining chapter is Ecosystem. So after this, we'll be entering into Ecosystem. So we can proceed, Cell, the Unit of Life, 7th one. And what all things we'll be discussing in this episode. For that, I will share the screen so it will be convenient for you. You can also find the notes. I think screen came, so we can proceed. Cell, the unit of life, 7th one. And main items, as usual, there will be an introduction. Then controlling unit of a cell, that is nucleus. Inside the nucleus, chromosome. Then 5 theory question, 5 NEET model question with the key. Remember, in shorts, daily I am putting 3 questions. Gradually will increase 2 plus 2, 2 first year and 2 second year. One set will be in between 12:45 to 1:30 in the afternoon time. Then other half will be after 8:30 or 9 p.m. So to get all this, subscribe the channel Ace Botany. Already many questions I put. So that will be there in Ace Botany. So please subscribe the channel. If anybody wants, please share it. Now we can proceed. Introduction. In this episode, I will told you what all things we'll be discussing: nucleus, chromosome, and question, and we'll be continuing eukaryotic cell because prokaryote no nucleus. Now nucleus. Concerned with the nucleus, many scientists are involved, of which few names seems to be more important. Just giving. First observed by Leeuwenhoek. Then nucleus as a cell organelle, explained by Robert Brown in 1831. And the material studied was in orchid roots. Now, W. Fleming, his work is the nuclear material stained by basic dyes was given the name chromatin. So the term chromatin after staining with the basic dyes given by W. Fleming. So important question for us, 1879. Normally, nucleus contains all the genetic information. That is why I told you controlling unit of a cell, eukaryotic cell. Nucleus is the largest cell organelle. Now there is the term interphase nucleus. So what is meant by interphase nucleus? So interphase nucleus means that is a nucleus. I think first page is clear. We can proceed to second page. So interphase nucleus means a nucleus in the non-dividing or metabolic phase. Nucleus is seen in all eukaryotic cells. So that is why it become eukaryote. Nucleus is there. But 1 or 2 exceptions are there, except mammalian RBC and sieve tube cells in plants at maturity. Now, next aspect is how many nucleus in a cell, that is number of nucleus. So nucleus, there is a technical prefix. It is called karyo or karyon. It's almost same as nucleus. That is why in cell cycle we studied karyokinesis, nuclear division. Number of nucleus can be 1. It can be 1 plus 1. A term we'll be studying in fungus, etc., dikaryon. Then 2, like certain organisms like paramecium. Then many, cynocytic, syncytium, rhizopus, etc. Then nucleus absent, that will not come here since we are dealing with the eukaryote. If nucleus absent, it is a karyote. So normally 1, 1 plus 1, 2, many. Now structure of nucleus. A typical nucleus consists of following parts. That is what? Broadly accepted, specifically connected with NCERT, nuclear envelope, nucleoplasm, nucleolus, and chromatin. Now, one by one, we have to proceed. First is nuclear envelope, which has got some synonyms like karyotheca, nucleolemma, all are synonyms. And this envelope, normally envelope separate inside and outside in any case. Here also, this envelope separates the cytoplasm and nucleus. And there are 2 membranes in connection with the nuclear envelope, like mitochondria, outer and inner. Generally, inner membrane is smooth, while outer membrane can be also smooth, or it is cytoplasmic part may have ribosome. So outer membrane can be smooth, or cytoplasmic parts may have ribosome. Outer membrane have connection with the endoplasmic reticulum. I think membrane is clear. Now we can proceed to next page. Next is perinuclear space. Perinuclear space means gap between 2 membranes, wherever it comes. So space between two membranes is called perinuclear space. It is 10 to 50 nanometer in width. NCERT data can be a question, theory fill in the blank. Thus, this gap may act as a barrier between inside and outside. At number of places, nuclear envelope is interrupted by minute pores. That is called nuclear pore. Now, first diagram, this one, old NCERT. This is a typical diagram. It is drawn diagram. So it will be clear. Labeling, everything is clear, all the parts, etc. Now we can proceed to next one. So in nuclear membrane, gap or perinuclear space, it can be 100 to 500 Armstrong. 100 to 500 Armstrong, or we can say 10 to 15 nanometer. In Armstrong, 100 to 500. Nanometer, 10 to 50. Nuclear pores, how it has been formed. So nuclear pores are formed by the fusion of two membranes. Through the nuclear pore to inside and to outside. Passage will take place both directions. And chemicals like proteins, ribosome, RNA. Now there is a structure called annulae, directly not given in NCERT, but a previous question. Annulae are circular structures around pores. The pores and their annulae form a pore complex called annulated pore. An annulated pore may have 9 cylinders arranged 8 plus 1. So the last area not directly given in NCERT since it came for some previous exams. Just to mention it. Now comes next part. It is nucleoplasm or nuclear matrix or karyolymph. It is comparatively transparent. It is colloidal. It is semi-fluid in nature. Fill the nucleus and it contains nucleolus. Highly extended nucleoprotein fibers, chromatin also same. I think that is clear. Everything labeled here, so it will be easy for you. All the parts. Now, nucleolus. See, nucleolus, there are spherical bodies found in the nucleoplasm. Nucleolus, remember, it is amembranous, like ribosome, like centriole. Nucleolus is also amembranous. It's a common question. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. And nucleoli in their turn consist of 4 parts based on the nature. The sense amorphous parts, granular parts, fibrillar parts, and chromatin parts. Connected with the nucleolus, 3 famous scientists. Many are there here. 3 names I just mentioned. Mainly scientists say that the initial discovery, etc., connected with the scientist called Fontana. And description is by a scientist called Wegener. The name connected with a scientist, Bowman. And what are the functions of nucleolus? It is the main site for the development of ribosomal RNA. It is the site of formation for the ribosomal components. And it can also act as a storage organ, specifically nucleoproteins, connected with the spindle formation and cell division. Now comes next material part. It is chromatin. So what is chromatin? It is the DNA protein fibrillar complex. Connected with the hereditary, chromatin can be stained by basic dyes. The name given by Fleming in 1879 as chromatin, we studied in the beginning. And they form a network-like structure called chromatin reticulum. Diagram, it is clear. Chromatin fibers are distributed throughout the nucleoplasm. Here you can find it. Nucleoplasm. And they are differentiated into 2 regions, euchromatin and heterochromatin. That part you will be studying in genetics second year. Euchromatin and heterochromatin, genetically active, genetically inactive, which will take more stain, less stain, etc. Now comes next component for us to study is chromosome. Chromo, wherever it comes, color, chromoplast. So means body, thus colored body. Again, here also many scientists involved. So initial works are concerned with the famous scientist Walter Fleming. And Heinrich Waldeyer given the term chromosome. Many scientists are in each cell organ. Few names just mentioning. Specifically, if it is given in NCERT, better by heart. Now we can proceed to next page. This diagram, that is structure and organization of chromosome we are proceeding. This diagram belongs to old NCERT. And this representation, that second one, this one, it is called ideogram of human chromosome, old NCERT, diagrammatic representation arrangement of chromosome, ideogram. And chromosome normally rod-shaped thread-like stainable condensed chromatin fibers. They are chromosome. And different stains can be used. Presently, for a common use, most commonly used stain is acetocarmine. Now, photographs of chromosome, then different types of chromosome, NCERT. Now, chromosomes are hereditary carriers, and they are clearly visible during karyokinesis that we studied in cell cycle. That is, shape is clear in which phase, chromosome will be more clear in which phase, etc. Now, chromatin contains what all things chromatin may contain. Contain DNA basically. Then some basic proteins called histones. What is all basic proteins called histones? And some non-histone proteins. Also RNA. That is what given in NCERT page number 101. Number of chromosomes in a species is usually constant. So a typical example in humans, in diploid cell, it is 46 to 1. While diploid cell means body cells, somatic cell, 46 to 1. When gametes are formed, it becomes 23, that is N, haploid due to meiosis, 2N to N. Now, structure of chromosome. When we look on to structure of chromosome, here I will explain. Central constricted area, this part, it is called centromere, and that is supposed to be primary constriction. On the side of it, that is not clear here, central constriction only we can find, centromere we can find. On the side of it, there is a disc-shaped structures. There is disc-shaped structures seen, and they are called kinetochores. It's a famous question. Disc-shaped structures, kinetochores, that cannot be seen in this diagram, NCERT diagram, kinetochores. And what is importance of it? Spindle fibers attaches during anaphase in cell division. And based on the position of the centromere, different types of chromosome based on shapes can be seen. NCERT diagram, very famous question, everything I marked there. So we can proceed to next page. So this is metacentric. That is, centromere will be at the center. Shape will be V. V in the sense to both sides it is equal arm. That is why the term letter given V. Both arms are equal length, V. Centromere at the center. Next is submetacentric, near to the center. So one arm will be comparatively longer, so it looks like letter L, capital L shape. Then acrocentric, near to one end, so it looks like the letter alphabet, capital J. And telocentric, that is at the terminal part. So it looks like letter I. So VLJI, center, near to one end, at terminal. Metacentric, submetacentric, acrocentric, telocentric. It's a theory question. It is an entrance question, usual question. Now, even diagram-based question. End of the chromosome. End of the chromosome is usually called telomere. Telo means end, telophase. Cell cycle will be remembering, telomere. Some chromosomes have additional constriction besides the primary constriction. Here you can find. So secondary constriction you can find. It's labeled there. Secondary constriction. So additional constriction besides the primary constriction. Such chromosomes have non-staining NOR. What is NOR? Nucleolar organizing region. That is somewhere here in this junction. NOR. Secondary constrictions at a constant location. This gives the appearance of a small fragment. Here you can find a small fragment. And that small fragment is called satellite, otherwise SAT chromosome. And it's important in genetic engineering or molecular basic genetics, we'll be studying it in fingerprinting, etc. Satellite, what is importance of it, SAT chromosome, etc. In humans, satellite chromosomes are 5 pairs, important one, 13, 14, 15, 21, and 22nd chromosome. 5 pairs of satellite chromosome. I think idea clear. Now we can proceed to next page, the chromosome, everything labeled here, NCERT diagram. Now, next is Lamprecht chromosome. This part not in syllabus, but some giant chromosome, just for namesake. Lamprecht chromosome concerned with the scientist Ruckert. Notice when primary oocyte nuclei, oocyte means egg, it is present in vertebrates and invertebrates, specifically diploid bivalent chromosome. Another giant chromosome is called salivary gland chromosome, otherwise called polytene chromosome. This is concerned with the scientist E.G. Balbiani. It is present in salivary gland cells of order Diptera insect. That is why it is called salivary gland chromosome. Numerous threads will be there, so polytene chromosome. And these two are exceptional chromosome, giant chromosome, not in direct NCERT, but just to mention for a namesake. At least you can eliminate when this options comes. Now, after that, when we proceed next to page, 5 theory question, 5 NEET model question with the key. So, what all things we learn in episode? For that, we can just rewind it. Comparatively a smaller one, but exam point of view important. Because nucleus, chromosome, etc. important for exam, second year and first year. So we started with introduction, what all things we'll be learning. Then nucleus started, who first observed it and who identified it as a cell organ, Robert Brown 1831, orchid root, then W. Fleming, that is term chromatin given, then with the staining with the basic dyes, 1879. Now, importance of nucleus is the master unit of a cell containing all the genetic information. The largest cell organ, interphase nucleus means a nucleus in non-dividing or metabolic phase, that is interphase nucleus. Then, nucleus is usually seen in all eukaryotic cells. There only we can find it exception, mammalian, herbacean, sieve tube cell at maturity. Number of nucleus, normally 1. It can be 1 plus 1, dikaryon. It can be 2, diploid. It can be many. 0 means not seen in eukaryote. It is then eukaryote. Now, structure of nucleus. When we say structure of nucleus, say when we say 0 nucleus means better use the term prokaryote. No nucleus and well-developed cell organ. There is a group called eukaryote viruses. So better use a prokaryote. We cannot find any well-developed nucleus. Now comes structure of nucleus. 4 parts: nuclear envelope, nucleoplasm, nucleolus, chromatin. And what are the synonyms for nuclear envelope? 2 membranes. What are their speciality? Then gap between 2 membranes, it is perinuclear space with a gap of 10 to 15 nanometer or 100 to 500 angstrom. Act as a barrier. Then a nuclear membrane has got some pores, minute pores, that is called nuclear pore. Diagram you can find here, so it will be easy. Nuclear pore. And when we come to nuclear membranes, again, space is 100 to 500 angstrom. Here, this you can find. Then nuclear pores are formed. How it has been formed by, that is formed by the fusion of 2 membranes. Certain area they get fused, so gap will be formed. And through nuclear pore, inside and outside conduction takes place for the materials like proteins, ribosome, RNA. And there is a special structure called annulae. Then annulated pore. Then total annulae connected with the pore, 9 cylinders, 8 plus 1, that part not in NCERT, just mentioned. Then nucleoplasm, nucleomatrix or karyolimb, transparent, colloidal, semi-fluid in nature, fills the nucleus. It contains nucleolus, highly extended nucleoprotein fibers, chromatin. Now nucleolus, what are the features of nucleolus? Spherical bodies found in the nucleoplasm, a membranous, larger and more numerous nuclei are present in the cells actively carrying out protein synthesis. It's an important question, theory question. Now parts of nucleus, 4 components: amorphous, granular, fibrillar, chromatin, based on the nature. Then 3 famous scientists, many are there, 3 famous scientists connected with the discovery. Main scientist is Botany. Connected with the description, main scientist is Wagner. And connected with the name, main scientist is Baumann. Now functions, concerned with the development of ribosomal RNAs, site for the formation of ribosomal components, storage organ, nuclear protein, connected with the spindle formation, cell division. Now coming to next page, chromatin. So DNA protein fibrillar complex, stained by basic dye, name given chromatin by Fleming, 1879. Network-like structure, chromatin, they form a chromatin reticulum later. Chromatin fibers distribute throughout the nucleoplasm, differentiated into euchromatin and heterochromatin, that will be studying second year. Now comes chromosome, the meaning of the term, and scientist Walter Fleming, initial work, Heinrich Waldeyer given the term, then a famous NCERT diagram, including karyotype. Then the stain, it can be stainable, chromatin fibers, and most commonly used stain is acetocarmine, given here last, acetocarmine. Now, shapes of chromosome and parts of the chromosome. Chromosome are hereditary carriers. Then clearly visible during karyokinase, only that time only it is visible. And it contains, chromatin contains DNA, some basic proteins called histones, and some non-histone proteins and RNA, NCERT. Number of chromosomes is constant for a species. Human, it is 46 in diploid cell, that is 2n equals 46. And in gametes, when formed, it becomes 23 through meiosis, then it becomes haploid. Then central construct area of a chromosome is called centromere, primary constriction. Side of it, we can find discrete shape structures called kinetocores. Spindle fibers attaches during anaphase in cell division in kinetocores. Then based on the position of the centromere, different types of chromosome, like a famous question, NCERT diagram-based question, almost in all NEET exam, you can find this question, theory also. First is metacentric at the center, so shape become V. Submetacentric near to center L. Acrocentric going towards the end, that is J. And telocentric at the terminal I. Telo means end. Ends of the chromosome is called telomere. And some chromosomes have additional constriction besides the primary constriction. Here you can find. And such chromosomes have non-staining in connection with the nuclear organizing region, secondary constriction at a constant location. That gives the appearance of a small fragment projecting. It is called satellite, sat chromosome. And in humans, satellite chromosomes are 5 pairs, 13, 14, 15, 21, 22. Then 2 giant chromosome, Lamprecht chromosome, and polytene chromosome, scientists involved, etc. That part not in NCERT, but just mentioned. Then after that, 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe or like it, share it, comment on it. We can meet later. So take care. Bye. Remember, shorts question I am daily putting. 1 or 2 will be around 12:45 to 1:30. Other will be after 8:30 or 9 in the night. Thank you. For time being, we can wind up.